Hello everyone, in this video we will talk about transposons, okay, or transposable elements. Transposons or transposable elements, or they are also known as jumping genes. Okay. Transposons are found in both prokaryotic and eukaryotic organisms, and they are called jumping genes because they are able to move randomly around the same DNA, jumping from one location to the next, like this one. See, I will give an example. Suppose this is your transposon, okay? This is your DNA where your transposon is located, okay? So this region, if this is your transposon, this can get excised from the original location like this. This get excised, okay, and can bind or get inserted into a new DNA or in the same DNA at a different site, okay. Suppose this is a different DNA, okay, I'll give different color for that. Suppose this is another DNA and it will get inserted in this location with the help of some enzymes, okay. Into new location. So what transposons are? These are a class of genetic elements that can jump to different location within a genome, okay. Their existence was first inferred by Barbara McClintock in 1983. McClintock in 1983. Transposons, I have already told, they are found in both prokaryotes and eukaryote. In human genome, they comprise about 45% of the genome. Okay, this is a huge amount. And the process by which sequences are copied and inserted into a new DNA molecule, this process is known as transposition. The process of jumping, okay, from one location to another is known as transposition. There are basically two classes of transposons, like one is called your transposon and the other is called retrotransposon. The transposon, but the mechanism by which transposon genes jump from one gene to another is known as conserve process, and the mechanism by which retrotransposon jump from one gene to another is known as replicative process. Okay, this process is nothing but a cut and paste process. Cut and paste. Why? Because in this process the donor DNA, suppose this is your donor DNA, okay, suppose this is your transposon, okay, and this is your donor DNA. The transposon will have a gene for encoding the enzyme transposers, okay, transposers. That is actually this transposers enzyme is actually able to bind to the 3' end and 5' end of the transposon and then cut this transposon by forming a structure like this. I will show it to you. It will bind like this. Suppose these two are your transposes. They will bind to the 3'. Actually, they recognize the terminal inverted sequence that are usually present in a transposable element, and they bind to these inverted sequences like this, and then they form a dimer like structure like this. Okay, sorry, different color. I will use the other color. A structure like this will be formed. Okay, and your transposers will form a dimer. They will dimerize, and then results in cleavage. Okay, in this portion. Okay, and they will excise this part of the transposon. This part will be excised and will get inserted into a new DNA molecule. Suppose we take the color, this one. This is your new DNA molecule. Here your transposers will get inserted. With the help of this transposers enzymes, the transposon will get inserted in a new site, in a new DNA, or maybe in the same DNA. Okay, this is your target DNA. This is your target DNA. During insertion process, actually some nicks are formed here and here, okay, due to the transposers enzyme. So this nick is filled up by the enzyme ligase, and because of this some direct repeats are formed in the right and left side of a transposable element. Okay, wherever there is a transposable element get inserted into a new site, there will be formation of some direct repeats. Okay, and these are actually not part of the transposon. They are left behind whenever the transposable element leaves the DNA. Okay, this is the idea of conserve process, that is the movement of transposable elements by cut paste method. Okay, the second type is your retrotransposon, and they jumped from one locus to another locus by the process of transcription. They first transcribe the DNA into mRNA. Suppose this is your the DNA. This is your transposable element here located in this DNA. This is your donor DNA. What they will do, they will first transcribe into an RNA molecule, and with the help of the enzyme reverse transcriptase, it will be converted into cDNA molecule, and this cDNA molecule will now get inserted into a new position in the same DNA or in a different DNA. This is your target DNA, okay, or target site, you can say. So this is a copy paste method, because your transposable element is transcribed or copied to form cDNA, which will get inserted. It is not cut from this site. Okay, so this copy of this transposable element get increase in number because of retrotransposition. So what we can say, in conservative transposition, there is change in position of the transposon without increasing the copy number, because this part is cut and then paste in a new position. This DNA will be like this without any transposable element. Okay, this portion will be removed from this DNA, and there will be no change in the copy number. This remains the same. But in case of retrotransposon, this part is not cut. Okay, it will remain in the old DNA, but get inserted a new copy of the same transposable element. This will be, and this one and this one will be same. This one and this one will be same, but it's a copy of that original one. So there is an increase in number of the transposable element. So now we will see what is the parts or what is the composition of this transposon. Okay, usually there are some differences in eukaryotic and prokaryotic transposons, but normally these things are common in all type of transposable element. Okay, suppose the presence of a gene coding for transposase enzyme, which is the key enzyme because it is required for cut paste or insertion of the this transposable element into new DNA. So there will be a gene coding for transposase. Then there will be some inverted repeat sequences in the 3' and 5' end of this transposable element. Okay, next to the gene coding for transposase. This inverted sequences are nothing but reverse complement of each other. Suppose if this is CCTAA, this will be your TTAGG. Okay, reverse complementary in a reverse direction. The transposase enzyme is able to recognize these inverted sequences. So your inverted sequences are transposase binding site. Okay, these sequences, your inverted repeat sequences, are your binding sites for transposase enzyme. That means what? Transposase enzyme will bind to the specific site of inverted repeat sequences, and then they will carry out the excision part. Okay, so this is about transposable elements. So if there is one thing I have already told, that there is some repeat sequences are formed. These are called direct repeats. They are formed because of the duplication of this base sequences after elimination or insertion of the transposable elements. So presence of direct repeats, suppose these are two direct repeats. If you can see direct repeats like this in a DNA molecule, you can say it gives you a clue about the presence of transposable elements, elimination of a transposable elements from this site. Okay, if somehow there is any kind of mutation in your transposon element, suppose no transposase gene is formed. Suppose no transposase protein is formed due to mutation in the transposase gene. Okay, so what will happen? Your transposable element will not be able to move, because if transposase is not formed, then it will not bind to the inverted sequences. So there will be no excision. Same is the case if there is mutation in the inverted sequences, then transposase element, even if it's formed, it won't be able to recognize the inverted sequence if there is a mutation here. So your transposon will not be able to move. So whenever there is mutation in the inverted repeat sequence or transposase sequence, there will be no transposition. But in the lab, researchers can do one thing. Even if there is mutation in the transposase gene, if your transposase enzyme is not produced, but your inverted repeat sequences are functional, they are not mutated, then what the researchers can do, they can add some specific transposase that can bind this inverted repeat sequences, and they can help this transposable element to move from one location to another. That means what? There are, you can say there are two types of transposon: autonomous transposon, and non-autonomous. Autonomous one will have the intact transposes, which will work functionally, which will be functional, and that will bind and result in accession and insertion. Okay, but in case of non-autonomous, the gene will have mutated transposes, so no transposes will be formed, but they can function. Non-autonomous one can function, they become active, and they can jump from one location to another if a transposes enzyme from different or outside is added. Is it clear? So this is all about transposons or transposable elements or jumping genes. Okay, nowadays researchers are giving much emphasis on studying transposons because of their genetic variability in different species or even within a species, which give a clue about evolution. So researchers are giving much interest in studying transposons. They have a lot of research scope. I have made 3 videos of transposons. In my next video, you can see the eukaryotic and prokaryotic transposons. Okay, you will be able to see what are the types of transposons found in prokaryotic cells and the types found in eukaryotic cells, and what are their differences. Okay, I'll discuss about that in my next videos. Thank you for watching. Bye.